Okay, so welcome to this next video in which we're discussing the sinoatrial node. So we've now discussed the incredibly important role of the sinoatrial node in the cardiac cycle and in setting off the cardiac cycle. Okay, so what we now want to discuss is how do the cardiomyocytes within the sinoatrial node actually work? How do they generate these spontaneous action potentials? Because they will generate these spontaneous action potentials whether they have neural input or not. It's these cardiomyocytes, they have an intrinsic ability to generate an action potential rhythmically. Okay, so let's have a look at how. Okay, so is this enough space? Probably not. We'll get another piece of paper. Okay, right. Right, okay, so let's say we have a sinoatrial node cardiomyocyte here. Okay, right. Now, what happens is, let's say the electrical potential difference across the membrane of this sinoatrial node cardiomyocyte starts off at around -70 millivolts. So let me tell you exactly what that means. Let's make sure we know exactly what that means. So, if you have a little man sitting in the extracellular fluid, he can measure the electrical potential of the extracellular fluid, okay? And he'll get some number, which I'll denote E for electrical potential, and then I'll put subscript E to denote extracellular electrical potential, okay? And what that little man can do is he can move into the inside of the cell. So he can go into the inside, and he can do the same thing. He can measure electrical potential, so big E here, inside, okay? And he'll just get some number. Now, what he can ask is if he moves from extracellular to intracellular, how much does the electrical potential change? I.e., what is the electrical potential difference when you move from extracellular to intracellular? Now, the other name for an electrical potential difference is a voltage. So what's the voltage between the extracellular and the intracellular compartment? Just means what is the difference when you move from the extracellular compartment to the intracellular compartment in electrical potential difference. So this is going to be the new electrical potential, i.e. the electrical potential for the intracellular compartment, and then you need to subtract off what the original electrical potential was, which is the electrical potential in the extracellular compartment. So that is what the voltage across this cell membrane is. Right, now, let's say that initially it's around -70 millivolts. So what does that mean? That means that if you move from extracellular to intracellular, your electrical potential will go down by 70 millivolts. So this one, basically, is lower than this one by around 70 millivolts. Okay, right. So, now let's plot a graph. Okay, so if we plot against time the voltage from extracellular to intracellular, so voltage from extracellular to intracellular, then it starts off at this -70 level. Now, when the electrical potential difference across the cell membrane of a sinoatrial node cardiomyocyte is -70 millivolts, it's at this very low level, basically, is hyperpolarized, okay? Then what will happen is it will activate certain channels in the membrane of this cell. Okay, so let's draw one of these channels here. And this channel is known as an HCN channel. So this is an HCN channel, and I'll tell you exactly what HCN stands for in a moment. Okay, so I'm going to colour the HCN channel in blue here. Okay. Right, so now let's discuss this HCN channel in a bit more detail. So, the HCN stands for, okay, so this is our long name. HCN stands for the hyperpolarization activated and cyclic nucleotide gated. So, hyperpolarization activated and cyclic nucleotide gated. Okay, hyperpolarization activated and cyclic nucleotide which will be cyclic AMP, incidentally, cyclic nucleotide gated channel. Okay, so this is the cyclic nucleotide gated channel. Okay, so that's its big name. So where did they get HCN from? Well, the H came from the hyperpolarization. The C came from cyclic, here. Okay, and the N came from nucleotide. So hyperpolarization, cyclic nucleotide is really where this HCN came from. Okay, now what is the structure of these HCN channels? Well, if we draw the structure of one out in a bit more detail, let's draw it largely here. So it's a channel. Okay, so here is a cylinder drawn in the membrane of the cell. So here's the membrane of the cell. Okay, and here's the pore through which the ions are going to move. And basically it's made up of 4 pieces. So I'm going to divide it up into 4 because it is a tetramer. Okay, so the HCN channel is made up of 4 separate proteins that are stuck together. Now, there are 4 different HCN genes in the human genome. So there is the HCN1 gene, there is the HCN2 gene, okay, there is the HCN3 gene, and there finally is the HCN4 gene. So these are 4 genes in the human genome which all code for HCN proteins. So we have an HCN1 protein, an HCN2 protein, an HCN3 protein, and an HCN4 protein. Okay, now these proteins, they make up a quarter of the HCN channel. So each one makes up a quarter of the HCN channel. So what has to happen is you have to make 4 HCN proteins, and then you can join them together to make an HCN channel. So the HCN channel is a tetramer of HCN proteins. So these are the HCN proteins. Okay, now the next question is, when you make a channel, do you have to use the same HCN protein in all 4 slots? Well, you can do, and you don't have to. So if you do use the same HCN protein in all 4 slots, it's known as a homotetramer. Okay? If you don't use the same HCN protein in all 4 slots, then it is instead known as a heterotetramer. Okay, right. So, now, let me tell you about the structure of these different HCN proteins, because they have very similar structure to the structure of the voltage-gated potassium channel proteins. So, each one of these 4 subunits which makes up the HCN channel here, so this is the HCN channel, made up of a tetramer of HCN proteins. Now we're looking at an HCN protein and how it straddles the membrane. So what you'll have is you'll have the amino terminus of the protein here. Okay, then you have the first membrane spanning portion, then the second membrane spanning portion, the third membrane spanning portion, the fourth membrane spanning portion, the fifth membrane spanning portion, the P-loop, the 6th membrane spanning portion, and then an important point here where cyclic AMP binds. So this is the nucleotide binding domain here. So I'll put this, label this, because this will be important later when we come to discuss how beta-adrenergic stimulation increases the heart rate. Okay, so this is the nucleotide binding domain. Okay, right, so let me colour this in to make it look a little bit more interesting. So we'll colour it in blue because we'll stick with this convention here of a single subunit being in blue. So this is all one HCN protein. So it has, this is the polypeptide basically. So here's the amino terminus, and here's the carboxyl terminus over here. Okay, so it's just a polymer of amino acids, and it's encoded by a single gene and made by a ribosome. Okay, and here it goes. And it straddles the membrane like this. So it straddles the membrane 6 times, and it also has this P-loop where it attempts to straddle the membrane but doesn't manage it. And an important intracellular domain is this nucleotide binding domain, which I've drawn here. Okay, so to label up the different membrane spanning portions. So, each of these membrane-spanning regions basically has a name. So the first one is known as S1. The second one is then known as S2, so that's this one here. Third one is called S3, so that's this one here. Okay, then the next one is S4, so the one I just traced, this one here. Then S5 here, okay. Then, oh whoops, not S4 again, S5. Then you have this P-loop here, so this portion that tries to straddle the membrane but doesn't manage it, that's the P-loop. And then finally, this portion here, this final bit after the P-loop that does straddle the membrane is S6, sorry. Okay, so you have these 6 membrane-spanning domains, and then this P-loop also. And basically, what happens is 4 of these proteins come together to make the whole HCN channel. And what happens is that the P-loop here will line the pore of the channel. So if you are an ion moving through this channel, what will happen is if you come in from the extracellular aspect, initially the portion of each of these proteins that you'll be in contact with will be the P-loop. So you'll be next to the P-loops of all 4 of these subunits, okay? Then, as you move further through the channel and you get towards the intracellular aspect, you can't be in contact with the P-loop anymore because the P-loop doesn't make it all the way through the membrane. So it stops lining the channel, and instead the bottom portion of S6 here takes over. So further down in the intracellular portion of this channel pore, you will be next to the S6 domains, the lower S6 domains of all of these subunits. Okay, another important fact is that it's the S4 domain, so this 4th membrane-spanning region here, which actually is involved in sensing the voltage and reacting to it. Okay, right, so which HCN channels are in the heart? Well, this is where it gets difficult. We can say which HCN proteins are in the heart, because all we have to do then is just do a basic assay where you work out how much mRNA for each gene is in the cell, and you can do that. Whereas actually seeing how they oligomerize together, how they tetramerize together, is more difficult. So I can tell you which HCN proteins are in the heart, and you can then imagine which HCN channels that will be in the heart. Okay, so the HCN proteins that are in the heart, the main one, the one that is expressed hugely in the sinoatrial node, is HCN4. Okay, so HCN4, you have a massive expression of this protein in the sinoatrial node. Okay, you then also have little expression of HCN1 and HCN2. So you can imagine that most of the HCN channels you will build in the sinoatrial node are just going to be tetramers of HCN4, i.e. homotetramers, where you have 4 HCN proteins joined together to make a HCN channel, which is a pure homotetramer of HCN4. However, you might also get some heterotetramers, i.e. some mixes between the different HCN proteins where you use different isoforms of HCN in each of these slots to make a heterotetramer HCN channel. Or you might even get some homotetramers of HCN1 and HCN2. So there's a variation of the different HCN channels that are in the cardiomyocyte, but mainly they're probably going to be HCN4 homotetramers. Okay, right. So now we've discussed the structure of the HCN channel. Let's discuss what it actually does. So, it's activated by hyperpolarization, basically, which is odd. Most ion channels are activated by depolarization, by the electrical potential difference across the membrane going up. These are activated by the electrical potential difference across the membrane going down by hyperpolarization. So when the voltage across the membrane gets to hyperpolarized regions like -70, these channels open. Okay? Now, what are they permeable to? Well, they're permeable to 2 ions roughly equally. They are permeable to potassium ions, so they are permeable to potassium ions, and they're also permeable to sodium ions. Now, let's try and think this through. Which of those is going to be more important when we have this cardiomyocyte at negative 70 millivolts? So, let me give you the figures you need. The potassium concentration intracellularly is 155 millimolar. Okay? The potassium so this is the potassium concentration intracellularly. The potassium concentration extracellularly is 4 millimolar. Okay? The sodium concentration intracellularly is roughly 12 millimolar. And sorry, these should have all been roughly. It's not precisely to whatever number of decimal places that. It's roughly. Okay? The sodium concentration intracellularly is roughly 12 millimolar. And extracellularly, it's roughly 145 millimolar. Now, I'll give you some time to think what's going to happen when we open a channel that is permeable to both sodium and potassium, and think we are at negative 70 millivolts, which means that this electrical potential of the intracellular compartment is lower than the electrical potential of the extracellular compartment by 70 whole millivolts. Okay, so you try and figure out what's happening, and I'll see you in the next video.